Have you ever looked at someone and thought, there's no way they're 60? Or met a 40-year-old who seems to carry the weight of someone much older? You know, that gap between the number of candles on the cake and how old our bodies actually feel? Well, that's always been a bit of a mystery. But now, scientists are building incredible tools to turn that mystery into a precise measurement.
So, let's just picture this for a second. We've got two people, both born in the same year. They're both 50. Person A is, absolutely thriving. They're active, they're full of energy, but person B feels totally run down, maybe struggles with frequent illnesses. They have the exact same chronological age, but their bodies are telling two completely different stories.
And this really gets to the heart of it, right? How can we actually quantify this difference? How do we measure the speed at which our bodies are declining, completely separate from the number of years we've been alive? Because, answering that question is the absolute key to understanding, preventing, and maybe even one day reversing age-related disease.
All right, so here's our game plan for today. We're going to start by looking at the very first attempts at aging clocks. Then we'll dive deep into how a much, much better one was built. We'll check its accuracy, and then we'll look at what this all means for the future of our health.
Okay, so these first attempts to really measure biological age, well, they needed a huge breakthrough. Scientists had to find something in the body that changes consistently over a whole lifetime, kind of like the ticking of a clock. And they found it. Not in our genes themselves, but in the system that actually controls our genes. The answer they landed on is a process called DNA methylation. Now, the easiest way to think about this is like this. Your DNA, your genetic code, that's like your body's hardware. It's fixed. DNA methylation, on the other hand, it's like the software. These are these tiny chemical tags that get attached to your DNA, and they tell your genes when to turn on and when to turn off. And as we live our lives, the pattern of these tags changes, basically creating a living record of our journey and a way to track our age.
So these first-generation epigenetic clocks from scientists like Steve Horvath, they were totally revolutionary. But researchers pretty quickly hit a wall, a big one. See, these clocks couldn't really tell the difference between the slow, steady process of aging and the noise from an active disease. They also got confused by generational differences. The fact that someone born in 1940 had a very different life than someone born in 1980. They called these cohort effects. And maybe most importantly for any real-world use, they just weren't reliable enough. Science needed a new tool, a better one.
And this really set the stage for a new scientific quest. The goal was to build not just another clock, but a speedometer, a tool that can measure the current pace of your aging, and do it while filtering out all of that other noise. And this is the story of how they did it, the DunedinPACE recipe.
The solution they came up with was this really elegant four-step process. It all started with a dataset that is truly unlike any other on the planet. And then they used some powerful machine learning to solve the exact problems that had stumped those first-generation clocks. Let's walk through how they did it.
Step 1. You have to start with the perfect data. And they found it in the Dunedin study. This is this incredible, one-of-a-kind project that has followed over 1,000 people who were all born in the same city in the same year. Just by doing that, by studying a single birth cohort, they could instantly get rid of those messy generational cohort effects that flawed the older clocks. It created a perfect, clean laboratory for studying age. Okay, for step 2, they did something truly groundbreaking. Instead of just looking at people at one single moment, they tracked 19 different biomarkers for each person over 20 years. We're talking everything from cardiovascular health like VO2 max and blood pressure to metabolic and immune system markers. They measured this stuff when the participants were 26, 38, and again at 45. This allowed them to calculate one single powerful number for each person, their personal pace of aging. Basically, how many biological years were they aging for every single calendar year that went by?
Step 3 is where it all comes together. They took that pace of aging score from the real-world biomarkers, and they took all the DNA methylation data from their blood, and they fed it all into a machine learning algorithm. Its job was to act like a superpowered detective, sifting through hundreds of thousands of possible DNA methylation sites to find the very few that changed in perfect lockstep with a person's actual real-world pace of aging. It was looking for the unique fingerprint of aging speed.
And the result was just stunning. Out of almost 1 million possible DNA markers to look at, the algorithm found that it only needed a lean, incredibly powerful set of just 173. That's it. These 173 specific spots on the DNA were all that was needed to create a highly accurate biological speedometer from a single drop of blood. So they had built this new clock. It was elegant, it was based on amazing data, but was it actually any better? To prove it, the team then had to put DunedinPACE through a whole series of really tough tests to validate its precision and its accuracy.
Now, DunedinPACE was actually the second version of this clock.
Its predecessor, called DunedinPACE, was sort of the proof of concept, but PACE was designed from the ground up to be better. It was trained on 20 years of biomarker data instead of 12. It used 4 different measurement time points instead of 3, which makes it more precise. And this is really key. The scientists deliberately threw out any DNA methylation markers that were known to be noisy or unreliable to make sure the final instrument was as solid as a rock.
And the results really do speak for themselves. What you're looking at here is test-retest reliability. Basically, if you test the exact same blood sample twice, how close are the results? A perfect score would be 1.0. And as you can see, DunedinPACE scores an incredible 0.96. It outperforms its own predecessor and other top-tier clocks. This kind of rock-solid stability is what makes it the gold standard for things like clinical trials, where you absolutely have to trust that any change you measure is real.
Okay, so we have an accurate, reliable speedometer for aging. But so what? What does it actually tell us? Why does measuring this pace even matter for our health and our future? Well, it turns out it's a very powerful predictor of what's to come.
When researchers tested DunedinPACE in other huge health studies, like the famous Framingham Heart Study, it proved to be a powerful fortune teller. A faster score in midlife didn't just line up with how healthy someone was right now, it predicted their future. People with faster PACE scores were way more likely to develop chronic diseases, to experience disability, and even to die years later, completely independent of their calendar age.
But the creators believed a true pace of aging measure should do more than just predict the future. It had to reflect the past. It needed to be sensitive to our life experiences, especially the tough ones, the things that we know can lead to a shorter, less healthy life. It needed to tell the story of a person's entire life, written in their biology.
And what they found here was just breathtaking. When they looked at another big study, they found that by just 18 years old, individuals who had grown up with major adversity, things like poverty or violence, already had a measurably faster pace of biological aging. I mean, think about that. This is profound. It proves that the stress of our early life literally gets written into our biology, leaving these molecular scars that speed up aging long before any disease ever becomes visible.
DunedinPACE is a massive leap forward. There's no doubt about it. But it's also just the beginning. The exact same approach is now being used to create a whole new generation of tools, giving us an even more complete picture of how we age.
Seriously, the science here is moving at lightning speed. That same methodology is now being used to create aging clocks from brain scans and from the thousands of proteins floating in our blood. The ultimate goal here is to create a full dashboard for human aging, giving us this complete, real-time view of our own biology.
And all of this innovation, it leaves us with a really powerful and pretty personal question. If a simple test could tell you your body's true speed, whether you're aging faster or slower than the calendar says, would you even take it? And if you knew the answer, what would you do differently tomorrow?